Okay, today I think is one of the most fun lectures of the quarter, and hopefully you will also agree. So we're going to talk about how microbes contribute to food that we eat in a good way, and also how microbes can spoil our food in a bad way, and either in your refrigerator or even in the fields before harvest. And I have managed to lose my laser pointer, so sorry about that. Okay, so just starting our lectures, it's been known that humans have been utilizing microbes, whether they knew it or not, for food production for many, many years. Okay, so this can date back to 5,000 BC. There's evidence of this jar, this Neolithic jar that contained wine, so you can tell based on the carbon fingerprints that there was wine in that jug, and that had been fermented. Okay. Today, we also have industrial processes that we'll talk about that are where bacteria or other microbes are grown in large fermentation vessels, and we utilize that those processes that bacteria are undergoing, and we purify products from that to turn into commercial products that are used for our benefit. But first, we're going to talk about microbes as food. And if we have enough time at the end, I'll show this short video about mushroom farming in California. So we grow a lot of mushrooms here in the state of California. And I was researching that before this lecture. And there's actually a lot of them very close by. So there's some in Escondido, Murrieta, and other places. The video is actually from Sacramento, but there are these mushroom farms throughout the state, and some of them produce Agaricus bisporus, which is just your regular white button mushroom. So if you go to the grocery store and you buy the white mushrooms, those are Agaricus bisporus. And also another fun fact is if you are buying portobellos, it's the same species, it's just older. So you just would let them grow longer. So if you ever, again, are in some Trivial Pursuit type of situation, you have this nice fun fact. So you'll know that they are both the same species, just older. And they grow in this sort of soil-type matrix here in mushroom farms that actually do very well in industrial areas because you can grow them in warehouses. So you don't need a lot of sunlight. You don't need a lot of things that you would need to grow a crop such as a plant crop. Okay. Now there are other mushrooms that you've probably seen in the grocery store that you've eaten that are specialty varieties. So they're not the white button mushrooms. And these are your Asian varieties, like your shiitakes, your oyster mushrooms. And some of those have to be grown in complex with their host. Okay? And so they need to grow on wooden blocks. All right? So shiitake, for example. And if you want to, you can actually buy these mushroom growing kits for your house. You can buy like this wooden block, and it's seeded with mycelia of shiitake or something, and they will grow in the house. And another fungus that we utilize a lot is Saccharomyces cerevisiae. So there are different strains of that, and they're used for different things. The most common use is in bread making or in beer making, but also in wine making that we'll talk about. Okay, another microbe that we utilize for food are algae. Okay, so these are things that grow in the ocean and are seaweed, right? So a lot of these things we can grow in seaweed farms, as you can see here in the picture, and that will turn into the nori wrap that you use for sushi. So red algae of Porphyra species is what nori is for making sushi rolls. And then there's also another utilization for algae, which is alginate. So that's a thickener. It's a product or a carbohydrate that these algae produce, especially these macrocystis species. And we can purify that and use it as a food product to thicken things like ice cream or salad dressing. And then in general, so bacteria, we don't use them that often for like the primary food product like you would a mushroom, because there's a lot of their DNA. It has a lot of like too much DNA in proportion to the amount of protein, and that can be hard for us to digest. So, but there's one example, which is the cyanobacteria, which is called spirulina. And so if you go to a health food store, you've probably seen it on the shelf. You can buy it in capsule form. It's sold as a health food product, and sometimes put in smoothies and kind of gives it that greenish blue tint. That's spirulina. And it's labeled as saying that it's rich in vitamins and nutrients and antioxidants. And so that's why people just sprinkle it inside of things. You don't really need a lot of it. Okay, so now we're going to talk about, so that was like using the microbe itself as the food product. But here we're going to talk about using the microbial processes that these microbes impart on our food to benefit the food that the microbe is in association with. Okay, so we're going to talk about basically fermentation, and so that is, as a general definition, is treating food with microbes. Okay, so you either intentionally inoculate your food, or you let the natural microbiota within that system flourish, and then they will ferment the food. Okay, and why do we do this? So sometimes it's good to have the microbes digest the food first. It makes them more digestible for us, and so that's basically the same thing that's going on in your gut. These microbes are having a chance to break down the food so our systems can break it down easier. And a good example of that are soybeans. And so a lot of times soybeans are treated such that they are more digestible to us and they're treated with specific species of fungi. Other times we use it to preserve food. So we talked about acidic environments being very bad for bacteria in particular, because that messes with the proton motor force. If you have too many protons in the solution, the bacteria can't maintain the proton motor force and then they die. So you can intentionally make a food acidic by treating it with microbes that are making that environment acidic, and that kind of outcompetes the other microbes that might be trying to invade and spoil the food. Okay? And then the same thing goes with alcohol too. Alcohol is a very good disinfectant, so that's a byproduct of fermentation, and that's why beer and wine can sort of fend off other microbes from contaminating them. And we also know that bacteria in particular can make vitamins, right? So folic acid was one of our examples when we talked about antibiotic resistance and targets of antibiotics, and we can utilize that, those processes for benefit as well by having them make vitamins and supplying that, or supplementing our food with that. Okay, so we're going to talk about acid fermentation and alkaline fermentation processes and the foods that are the products of those. And the first example is Lactobacillus strain, XYZ, and there are 10,000 units of that in this cup of yogurt. The kefir, which is another milk product, kind of like a thickened milk product, is also a product of Lactobacillus fermentation. And there are some sausages that are fermented with Lactobacillus. Lactococcus is another organism that is used to make buttermilk or sour cream. And then Streptococcus, which we have learned about in terms of pathology, there are actually some beneficial strains that are used to make yogurt and kefir as well. And then others where are used for fermenting fish and other types of sausage as well. So that was all about acidic animal product fermentations. Now we're going to talk about acidic vegetable fermentation. And so I talked about this in the beginning, about soybeans. There are some elements in soybeans that are really hard for us to digest. And so most of the soy products that we eat, besides edamame, have been processed in some way. Okay, so that is treating with this fungus, tempeh, fungus rhizopus that makes tempeh, which is like a meat substitute, which is pictured here in the right corner, made from soybeans. Okay, and then there's Aspergillus species that we use to make miso and soy sauce, and through an acidic vegetable fermentation process. Now, soy sauce production, and also other things where Aspergillus might be prevalent, there are some Aspergillus strains that produce mycotoxins, which are very bad for us. They cause cancer. And so you have to make sure that you don't have Aspergillus flavus in your production line that's producing aflatoxin. Okay, so there's a lot of testing that goes on with soy sauce production. It can also contaminate peanuts because peanuts can be, they can acquire Aspergillus strains in the field. And so you have to make sure that your peanut butter pipeline doesn't have Aspergillus flavus in it. They have to do this routine testing to do that. You probably are familiar with kimchi. It is a acidic vegetable fermentation, okay? And it's basically pickling in high salt brine, okay? So that is putting these vegetables in a container with bacterium and a lot of salt and allowing it to sit and age for a long time, and then the product that comes out of that is kimchi. You're probably also familiar with sauerkraut, which is very sour, and that's because it's an acidic fermentation process that is contributing to that. Now we're going to move into alkaline fermentation, so not acidic, not low pH, but high pH. And this includes both animal and vegetable products. One of them is natto, which I've never tried, but this is fermented with a bacillus strain, and all of that stringy stuff that is connecting those soybeans together is the exopolysaccharide from the bacteria. And then dawa dawa is another example of fermented locust beans, which is via bacillus strain. So there are lots and lots of bacillus strains that are out there. We've talked about them in terms of anthrax and also biofilm formation with Bacillus subtilis. So there are some that are used in alkaline fermentation products. And then 1,000-year eggs are also an example of a bacillus fermented product that create these alkaline fermented, I believe they're duck eggs. Okay, so now we're going to talk about ethanol fermentation, which we're probably familiar with because we've all eaten bread before. And we probably all, at least at one time in our lives, bought baker's yeast from the grocery store to try and make our own bread. And what it's doing is, you know that you have to feed it sugar to make it activate, and it's converting that sugar into ethanol and CO2. So that's why you have these bubbles that are rising from that soup of sugar and yeast that you've given it. And once you mix that with bread and with flour and the other components of the bread, that CO2 causes that bread to rise. And then you bake it, and all the ethanol is baked off. And so that's why you don't have ethanol remaining in your baked bread. It's based on the ethanolic fermentation of Saccharomyces cerevisiae. And that's with wheat flour, okay? That's just our typical bread that we have in the United States. There's also another prolonged fermentation process from a grain that is not wheat-based, and that's called teff. And that doesn't have any gluten. It's gluten-free, and that's a typo, not gluten. Sorry about that. And it's called injera, and it's used in Ethiopian food quite often. And if you've ever been to an Ethiopian restaurant, you know they give you that very thin, like crepe-like material, and then you use that to pick up your food and eat it. And it's really, really good. I really like that. And it's fermentation process that gives us that, and it's dominated by the yeast Candida instead of Saccharomyces cerevisiae. Okay, so this different strain of Cerevisiae is used for making wine. Okay, so there are lots and lots of different strains of wine yeast, and there are particular wineries that ascribe to using, say, Pasteur Red or something, different strain of the yeast. So there are lots and lots of different strains, and some people have particular affinities to particular strains because they think it's going to impart a certain type of flavor into their wine. There's also forms of winemaking where you allow the natural yeasts that are on the berry skin to do the fermentation. Okay, so that's kind of a philosophical divide among winemakers of people who want just the natural microbiota to do it, or people who want really tight control over their fermentation processes and give that particular yeast to the grape juice before fermentation, so they know what the product more or less be at the end. Okay? And so wine is derived from the alcoholic fermentation of fruits, mostly grapes, but you can find other fruit wines that are out there, like apple wine and maybe you see like pineapple wine and stuff that really just doesn't sound good, but you can find it. And basically the whole process is crushing the fruit, in this case the grapes, to release the juice, and then either allowing it to ferment with the natural microbiota or intentionally inoculating it with your Saccharomyces cerevisiae strain of choice, for example, Pasteur Red, and then allowing that glucose to eventually be converted to alcohol through ethanolic fermentation. So that is with the yeast part of the fermentation process. A lot of wine also undergoes a second fermentation process, and that's called malolactic fermentation, and that will impart even more flavors. So a good example of that is like if you are listening to wine reviews or people talking about wine and they say that your Chardonnay has a buttery flavor, that's coming from this malolactic fermentation. It gives us sort of buttery undertones to wine, and that's actually from a bacteria, Enococcus emei. Okay, so this is how the winemaking process works. So on the left you have red wine, on the right you have white wine, okay? And it's slightly different based on if you're making reds or white. So obviously if you're going to make red wine, you're going to start with red grapes, okay? And you're going to crush them to get their juices out, and you're going to press the juices and the skins together, and then you're going to inoculate with yeast. You also inoculate with sulfur dioxide because that keeps the bacterial population down, because you do not want other bacteria that can get in there and contaminate and turn your wine to vinegar, okay? So you want to prevent that from happening. And then you allow that fermentation to occur by the yeast product. Then you need to chuck the skins, okay? You need to get the skins out of that mixture. And as you are fermenting, so you'll see that the skins rise to the top, and it forms what we call the cap. And you have to punch down the cap. Like you can actually do that with your hands. And then mix it all back together. Then the next day the cap will rise. You have to do that, probably for several times, maybe a week or so, that you have to keep punching down the cap. And then you're going to press it through a press, and you're going to get rid of the skins and just retain the juice. You don't want the skins anymore after that. And then you're going to allow it to continue fermenting just the juice without the skins. And then as that's happening, the yeast is dividing and growing and doing all the things that we talked about in terms of going into stationary phase and dying and all of that. And so you have to do what's called racking off the yeast lees. Okay, so you don't want a lot of dead yeast in the bottom. So you have to siphon off the wine from one vessel to another, and you leave the debris, which is the cell debris of the yeast, on the bottom of that vessel. And you have to do that a couple times. And then if you're going to do malolactic fermentation, you would add your Enococcus emei and allow that to happen. And then at that point you can age in an oak barrel if that's what you want to do. And then you can then go and bottle it after that and then sell it. Okay, so that's red wine. If you're making white wine, you immediately remove the skins. So the skins are not there. And the reason why you leave the skins in the red wine situation is because you want to extract all those anthocyanins and all those red pigments that are in the skins. Okay? You do not want to do that with white wine, so you just press the juice, immediately get rid of the skins, and then you do that fermentation process like I talked about. You have to punch down the cap and do all these things, and then rack off the lees and undergo malolactic fermentation if you want to, and then age if you want to, and then bottle it. And at all points in this process, you want to make sure that you're not getting contamination with acetobacter or other things that can turn the wine to vinegar. Okay, so beer is another example of ethanolic fermentation. So this again is a Saccharomyces cerevisiae strain that is used for making beer. And again, it's converting pyruvate to ethanol and CO2. And some of that CO2 is retained in the solution, okay? And that's why beer is bubbly. So that is gassed off when you're making wine, unless you're making champagne. But in beer, you're actually retaining some of that, okay? And it's based on the fermentation of grains like barley, wheat, and corn. And I actually saw, so there was like a Super Bowl ad, I think it was Budweiser, saying that they never use any high fructose corn syrup in their beer production. And I was like, like something they were proud of. And then I was like, oh, I didn't even know that anyone did that. And then there was like this sort of rebuttal article from the corn industry, kind of slamming them for saying that. Say, okay, well, this is a byproduct of our industry. Like, why are you trying to sell it as a bad thing? So anyway, I thought that was interesting. And I don't know if you remember that Super Bowl ad. We don't use any hyperfix scores. Okay, so that was all about making food with microbes and making our food taste better with microbes, or making microbes make some sort of beverage for us. But we also have to worry about microbes spoiling our food. One, because it causes them to taste bad, but also because you can get food poisoning if it's some sort of human pathogen, and you don't want that to happen. Okay, so if you have too much acidic fermentation where you don't want it to be, that can give you a sour taste. If it's alkaline fermentation, it can give it like a really nasty bitter taste. And microbes can also oxidize fat, which causes like rancid taste. So like if you have nuts that have a high fat content, you know what a rancid nut tastes like. It really does not taste good. Meat can become putrefied if it has too much microbial activity, and that is really not good as well. And then we all know that milk can spoil if our microbes get in there and turn it into some sort of acidic fermentation that we didn't plan on. We don't want to eat curdled milk. We didn't do it on purpose. I wanted to talk about seafood. Okay, so psychotrophic bacteria are ones that can live in the cold, okay? And they can produce this TMAO product, which contributes to the fishy smell. So when you bring them out of the ocean, they have these bacteria associated with it because it's a pretty cold environment. And that can, if you're buying a piece of fish that has a really bad fishy smell, that means it's not as fresh because those bacteria haven't been allowed to grow on that fish after it was harvested. So just keep that in mind. And then we also have a lot of problems with plant pathogens that can spoil crops in the field before we even harvest them. And we're going to talk about plant pathogens in the next lecture, I believe. And then we can also have human pathogens that contaminate food. So this is an example of peanut products that had this outbreak of Salmonella enterica, which is a diarrheal causing agent closely related to E. coli. And after doing a lot of genetic fingerprinting across the country, trying to figure out, if people in this state and in this state had some similar food that they had been eating, and also was the strain similar of the salmonella that they pulled out of these people, and trying to figure out and trace it back to where it came from. Okay, so they finally traced it back to a peanut product processing plant in Georgia, and it was not the actual peanut butter, but it had ended up in cookies and things like that, and so they were able to piece it together that all these different products that had this Salmonella enterica, actually it all was sourced back to this one peanut processing plant in Georgia that was sending out peanut butter to all these different types of cookie making companies. Okay, so plant food, so we have some post-harvest problems that can happen. So the plants or the fruit or the tubers or whatever it is that you're harvesting as the food product can actually acquire infections in the field that are not noticeable until after you harvest. And so that's called a post-harvest disease. So a good example of that is bacterial soft rot of potato. So you can harvest all these potatoes, package them, and send them out to market, and then they start to have this liquidy rot associated with them, and it's called bacterial soft rot of potato. And it's caused by these Erwinia species of bacteria that they actually have acquired during the growing process. And so that's not evident until you get them onto your grocery shelf, and their shelf life has plummeted because of that. There's also, we talked about in the very first lecture in terms of history of microbiology, shaping human history. And that would be this Phytophthora infestans, which is a fungal-like organism, an oomycete, that caused the Irish potato famine, and it caused a lot of immigration of people from Ireland and the United Kingdom to the United States. A massive movement of people that really helped shape the history of this country as well. Okay, so other foodborne pathogens that we might be aware of or might be interested in or might have heard of before. So listeriosis is a problem caused by Listeria monocytogenes. Okay, so this is again a psychotrophic bacteria, meaning that it can thrive pretty well in refrigeration, in cold conditions. So in places where you have refrigerated products, you might have contaminated, or there's a chance you could have contamination of Listeria monocytogenes. And this is particularly dangerous for things that we put in the fridge and we don't cook before we eat them. So examples of that would be deli meats and things like that. So they're already cooked, and then they're refrigerated, and they're packaged, and they're sliced, and then they're sold, and then we eat them. Generally, you're not going to cook your turkey or your ham before you put it on your sandwich. So there is a risk that they could be contaminated by Listeria monocytogenes. And this can be very bad for you, particularly for pregnant women. So it can lead to spontaneous abortions. And this is one of the reasons why OBGYNs tell their patients not to eat deli meats as well as unpasteurized cheeses because of fear of listeriosis. Okay, we talked about Clostridium botulinum a little bit when we talked about sterilizing techniques, and that this is a bacteria that can form spores, and so it can be hard to sterilize because the spores are very resistant to heat, and so they can be carried over sometimes in the canning process. Okay, so canning involves heating the cans to sterilize them, to effectively sterilize them, but if there are Clostridium botulinum spores in there, that can lead to botulism. Okay, and so that's a very bad disease. It causes flaccid paralysis, and it's because this bacterium is producing botulism toxin, and that is affecting how our nerves function. Okay. Now, one of the things that you might already know or you might find interesting is that Botox, which is something that people use for wrinkle control and also for treating some muscular diseases, is derived from Clostridium botulinum. It is one of the forms of one of the botulism toxins that has been used in a pharmaceutical product. But it came from this pathogen. Okay, so how do we preserve our food? Well, we can refrigerate it, obviously, or you can just eat it before it goes bad. So that's sometimes a problem we have in our household. It's like, oh, we buy these strawberries, and then, oh, look, now you have a bunch of botrytis on them, and now we have to throw them away. So we try not to do that as much as possible. Cooking is also a very good way. You know, ever since humans have discovered fire, we discovered that cooking our food is beneficial, makes it taste good, but also kills the microbes on it. We can pasteurize things. We learned about that in terms of heat treatment of mostly liquid products. And we can also use ultra-high temperature treatment, which sterilizes things and gives them a shelf life that doesn't require refrigeration. And then we can pressure cook, which is very similar to an autoclave for canning processes. And that generally will also kill resistant spores, hopefully. You can also remove the liquid water by freezing. Okay, so that's one way. You can also cure something by salting it, so like beef jerky and things like that are good examples of that. Or you can use one of those fruit dehydrators and dry all the liquid out of there. Or you can have a really high osmotic environment where you're supplying a lot of sugar. Okay, so bacteria and other microbes in general don't deal well under high osmotic pressure. So if you create that in your fruits and your jams, then you can kind of ward off organisms from coming in there. You can irradiate food, and so sometimes there are some food products where that is legal, and it's mostly for export. Okay, so other countries don't want our potential plant pathogens that might be, say, contaminating cherries or something that we had harvested here, so they ask that they be irradiated, and then they're shipped over to other countries. And then you can have organic acids that would reduce the proton motor force or disrupt it in some way. which does not do it justice, but I wanted to just expose you to it and maybe catch your interest in this in case you wanted to maybe look into this in terms of a career path at some point. And so this is basically using microbes to make products, and that can be vaccines, so we can have microbes that will produce components of a vaccine for us. They'll produce things like pharmaceutical products, and insulin is one example of a microbial fermentation process that we have engineered microbes to make insulin. Okay. Another useful compound from the actual bacteria itself is xanthan gum. We talked about that. Okay, so that is an organism that's actually a plant pathogen, and it produces loads of exopolysaccharides. This is like gooey, sticky substance. You can purify it away and put it in food products or cosmetic products. Okay. One of the workhorses of industrial microbiology is E. coli. There are also yeasts that are used as well. And so you can force E. coli to make what we call recombinant products. And so we talked about that. We talked about recombinant DNA biotechnology. And you can have them synthesize large amounts, depending on how big your fermenter is, of your product of interest. Okay? So it is important that you look into, so you have this protein, for example, that you're trying to make, and you see that it makes it very nicely in a flask, okay? But sometimes it's very hard to scale that up to really large fermentation processes. And so some things that look like they're going to behave very well for you, you try to scale them up, and then it like totally messes up the production of it, it produces less amounts of it, or somehow skews the production of that. So that's something to think about when you're trying to find something that you can turn into a product. And then we talked about recombinant DNA technology, so I won't go back into that. Okay, so this is an example of what a large fermenter would look like. Okay, and just imagine many, many, many of those in a particular biotechnology environment. Okay, so it's somehow, like I said, it's sometimes difficult to manage the microbes in the large fermenters because you have to keep in mind about their growth phase, and you don't want to end up with a bunch of dead cells that aren't doing anything. And so you need to make sure that they're constantly in the log phase, if that is in fact when your product is being made at its highest amount. You need to give it sterile nutrients, so broth. Okay, that can be hard if you're trying to make 10,000 gallons of microbial broth. That can be really hard to make sure that's sterile. And then you have to remove the product, get rid of all the waste. Okay, so at that point, the cell debris would be the waste. You don't want the cells anymore. And you need to purify that product. So these are all hurdles that you have to overcome when you're trying to create something from a microbe. Like, yes, it's great if it can make it, but then you have to be able to purify it. And through that purification process, it has to retain its properties. Say if it's an enzyme that we're putting into our laundry detergent to break down blood and other things, it needs to retain that activity throughout the purification process. We talked about transgenic plants and how we get those. Okay, so that would be mostly through agrobacterium-mediated transformation. And you can also have plants make things, either that would make them more resistant to disease, but also you can use them, plant cells, to make products. So you can transform plant cells, and they can make products just like a microbe could. Okay, so we have a few more minutes. I'm just going to show you that video of the mushroom farm, because I think you might be interested in that. And know that it's a huge industry in the States, and it's based on